corals are marine animals. Like all animals, corals eat, breathe, grow, and reproduce. They deposit hard skeletons that serve as the building blocks of reefs as they accumulate over thousands of years. There are three major types of coral reefs: atoll, barrier, and fringing reefs. Fringing reefs are the most common type of reef in the country. Coral reefs serve as a habitat for various marine life, supports food security, and provides shoreline protection to coastal communities. To protect and conserve these valuable resources, it is essential to know about the corals in a reef to formulate the appropriate reef management initiatives and strategies. An assessment determines the overall status and health of a reef, while monitoring detects the changes in the condition of crucial parts of the reef. Coral reefs are assessed every 5 years as stated in the Guidelines on Biodiversity Assessment and Monitoring System for Coastal and Marine Ecosystems. However, with the rapid changes in reef health due to climate change and human impacts, reefs must also be monitored once or twice a year and right after a significant disturbance such as mass coral bleaching or direct typhoon impact. A detailed and validated map of the reef area, including different types of corals present, determines the number and placement of the sampling stations in the assessment. Focus the assessment on fully formed reefs with 3 zones, namely the reef flat, reef crest, and reef slope. Conduct the sampling at the reef slope where the most variety of corals are found. Identify the locations for sampling in the area with these considerations. First, assign the reef slopes to 3 strata: the northeast-facing slopes, the southwest-facing slopes, and others, such as those in narrow channels or near the bay head. Second, allocate the number of sampling stations to these strata in proportion to their areas relative to the total area to be assessed. For example, assume a maximum of 10 sampling stations could be included in an assessment, and 60% of the total area of reef slopes faces the southwest, and 30% faces the northeast. 6 sampling stations must then be assigned to the stratum facing the southwest, 3 on the stratum facing the northeast, and 1 in a stratum classified as others. The position of the sampling stations must be determined randomly per stratum. We must remember that the number of sampling stations in an assessment is not fixed. Factors such as the size and variety of reef habitats and the availability of resources and expertise should be considered during selection. Repeat assessments should be done every 5 years. Monitoring stations are chosen from the baseline assessment sampling stations. Stations with high coral cover and diversity may also be selected or based on a specific monitoring objective. 15 to 25% of the sampling stations can be assigned as permanent monitoring stations. Monitoring is done at least once or twice a year or right after a significant disturbance, such as mass coral bleaching or direct typhoon impact. It is highly recommended that the monitoring stations are deployed in pairs, 100 to 200 meters apart. Each pair of monitoring stations is called a monitoring site. It is also recommended that there must be at least 3 monitoring sites. Assessment and monitoring of coral reefs must use the photo-transect method using 3 to 5 50-meter transects deployed parallel to each other at randomly chosen starting points within the 75-meter by 25-meter sampling station. Once the transect is laid out, digital photographs must be taken at 1 meter intervals. Use coral point count with Excel extensions, CPCE, to determine the abundances of reef organisms intercepted by 10 randomly positioned points in each photograph. To interpret the data, focus on hard coral cover and diversity. Unlike soft corals, hard corals contribute significantly to reef formation and are important in determining reef fish assemblages. Liquanan et al. 2019 introduced new measurement scales for interpreting hard coral cover and diversity. These scales allow for the development of adequate baseline and repeat assessments and monitoring that are more sensitive in detecting changes in reefs. This means better measurement of environmental impacts and effectiveness of management interventions. Coral diversity must be described using lifeform categories. Taxonomic amalgamation units, or TAUs, are prescribed for reef assessors with training in coral identification. 3 of the same 50-meter transects used for coral reef assessment and monitoring are used to collect data for the Fish Visual Census, or FVC. FVC is conducted to determine the composition of the fish assemblage in terms of density and diversity. Fish biomass describes the amount of fish in kilograms per square kilometer of coral reef. Identify all fish species found in the area and count all individuals present within the 5-meter belt along the transect. Divers may initially identify butterfly fish species and count individuals per species. Experienced reef assessors with expertise in fish identification should identify and estimate the sizes of the fishes per species, particularly the major and target taxa. Starting from one end of the line, each observer hovers on each side of the transect line while observing 5 meters on their side of the transect and move forward to the next 5-meter mark. Both observers swim and stop every 5 meters along the line to record the counts of fish per size and class until the entire transect is censused. Ideally, the status and composition of the reef fish assemblage are classified by the fish species as target species, major species, and indicator species. Identification of indicator butterfly species is used to determine the health and biodiversity of the coral reef, as half of the species of these fishes feed on corals. Compute the reef fish density by counting the number of fish individuals per 1,000 square meters. Compute fish diversity by identifying the number of species per 1,000 square meters. Providing the list of the top fish families and species will give a better view of the fish assemblage in the area. Fish length estimation is crucial in the measurement of fish biomass. Biomass is calculated as weight in grams equals the length of the fish in centimeters, raised to the power b, multiplied by the a value of the fish species. A and B values are unique to each species, and a list of these values can be obtained from the fishbase.org website. As shown on the screen, density and diversity are categorized based on the scale by Hilomen et al. Nanola et al. Is used to categorize biomass. Practice fish identification using a laminated field guide with top 10 to 20 species per protected area. Familiarize the different identifying marks unique to each species. Practice fish length estimation by deploying laminated images of fishes with predetermined sizes underwater. Our coral reefs and their associated coral and fish species contribute to food security and coastal resiliency, thus monitoring their condition and status to ensure that adequate and efficient methods are employed to protect and conserve these precious coastal and marine resources.